Good morning, students. Today we are going to discuss about product purification by ion exchange chromatography. It's a one type of column chromatography. So the major crisp introduction about the ion exchange chromatography I will be giving here. So normally in chromatography there will be two phases. One is stationary phase, another one is mobile phase. And the major principle of each chromatography will be varying. And here the stationary phase base material will be cellulose or agarose. It can be converted into a charged exchanges. Okay. In mobile phase will be of liquid. Mostly buffer system will be used in ion exchange chromatography. Then mechanism will be the in sample it will be in one charge, and in a column there will be an exchanger, ion exchanger. It will be of one charge. The opposite interaction between the stationary phase ion exchanger as well as the sample of interest. So that will be the major mechanism. Then advantages and disadvantages. One by one we'll go through now. Okay. So the first one is it is a process. Here, the separation of ions or polar compounds will be based on the affinity of the compound in the ion exchanges. So the major interaction will be ion exchanger compound as well as the sample of interest. Okay, so here the separation will be, it will be of mostly reversible exchange of ions between the target compounds and the ions present in the ion exchanger resin. Normally, two different types of ion exchanger will be used. One is cationic ion exchanger, another one is anionic ion exchanger. So, one by one, we'll see. What is cationic exchanger? Process negatively charged groups. So, it will attract positively charged cations. So, in that sample, if any positive, if our sample of interest is positively charged cations, so we can choose cationic exchanges in the stationary phase. In contrast, anion exchanges having positively charged group. So if it is positive, it will attract negatively charged anions. So if you want to separate negatively charged anions, the stationary phase will be packed with anion exchanges. So normally this ion exchange chromatography will be performed in column basis. So column chromatography will be only used. In some cases, thin layer type of chromatography will be also used. So here you can able to say, I have told no anion exchanger, cation exchanger. So this will be the example. So if you go through this, I have already told base material will be cellulose. Okay. So here CM cellulose will be there. So you can see carboxymethylated group will be added attached to the cellulose. So it will be of negatively charged. Okay. So it will attract positive charged compounds. So this will be called as cation exchanger. Next thing will be DEAE cellulose. That is DEAE. So you can see diethyl amino ethyl. So this compound will be attached in the cellulose. So it can be called as DEAE. So here it is positive charged group. So it will attract negatively charged compounds. So this can be called as anion exchanges. So what is the target of interest? Which type of target of interest we need to purify? So according to that, we need to choose the exchange resin in the column chromatography. Okay. So the working principle now we'll see. So here the major principle will be attraction between the oppositely charged in the stationary phase and the charges in the analyte. So in between these two only the attraction will be there. Okay, so the ion exchanges basically contain charged groups. This will be covalently linked to the surface of the insoluble matrix. Okay, so the normally insoluble matrix I have shown in previous slides, no, that is be cellulose. So that will be modulated. Either it is carboxyl methylated or DAE compound. Either it is positively or negatively charged. Okay, so this charged group of the matrix will be either positive or negative. So when the suspended aqueous solution, no, when these compounds will be suspended in aqueous solution, so the charged groups of the matrix will be surrounded by ions of the opposite charge. So this complete charged group we can call it as a ion cloud. Okay, these can be reversibly exchanged. So we cannot change the nature. or properties of the materials. Nothing will be changed, but these will be reversibly exchanged. Then instrumentation part, what are all the main components in the basic ion exchange chromatography setup? We'll see. Contains pump. Pump is injecting a mobile phase. That will be a buffer at specific flow. And injector. Injector is needed to for sample injection. Okay, so very lesser volume and we have to inject a sample. And a column. Column is used for packing of the stationary phase. And detector. Detector will be only used to find out which compound is eluting. Okay, then recorder and data system. These are all the instrumentation setup of this ion exchange chromatography. Okay, now coming to the working. So first, these ion exchange separations will be carried out in the column. So in the column, the ion exchanger will be packed. So the choice of this ion exchanger will be based on the component of interest we are going to elute. If we want to separate negatively charged. So according to that, what type of exchanger we have to choose. Previously, I have told no. So based on the component of interest, we have to pack the exchanger resin in the column. So first, the column is filled with the ion exchanger. Then sample will be injected, followed by buffer system. So in that sample, which will be having more affinity towards the stationary phase ion exchanger resin, so their attachment will be there. So other components less affinity towards will be lessly attached. So once you go through the buffer system inside, so which will have a less affinity that will come out first. So what type of buffer that can be used as a mobile phase here is tris buffer, pyridine buffer, acetate buffer, citrate, phosphate buffers. So these are all the buffers which can be used as a mobile phase in the ion exchange chromatography. Okay, so once you are running the buffer with the system, so the particles which will have the least affinity towards the stationary phase, that is the exchanger resin, that will come down first. So this step we can call it as washing step. Then we have to elute our component of interest, which will have high affinity, so which will tightly bound with the exchanger resin in the stationary phase. So here the high affinity towards the exchanger resin compared to this target of interest that has to be run through the system. Then we can elute our component of interest. Okay, so here we can tell no. So that process we can call it as a elution process. So these eluted compounds, no particles that can be analyzed spectroscopically. Then we'll run the column. Finally, we can regenerate the column with the help of HCl or NaOH ions because in the buffer some ions will be there elution, no, highly affiliated. That has to come out and regeneration of the ion exchanger resin is to be needed. Then it has to be gone for next step of the next type of purification step. Okay. Then the procedure I'll be showing in the simple diagram. Okay, so here you can see. So this is the matrix. So here the concentration of initial buffer, some buffer will be running no equilibration. So that will be surrounded here. Okay, you can, this is the gel matrix. So it is a positive charge in the exchanger resin. Okay, it will attract negatively charged. So then you have to run the sample. Okay, so the sample will be like different, see here triangle, two different colors will be there, no? So different charges will be there. So once you are once you are running the mobile phase here, no, buffer system, then you can able to see. So according to the charge of the matrix, the compound charge, the different charged compounds will be attached in the matrix. Then you are starting the elution. Okay, so while we are eluting, no, so highly affinity towards this exchanger resin, no, that is strongly attached. So less affinity compound here you can see, you know, this is a red color thing. This is less affinity towards this matrix, but this blue color compound, no, this will be highly affinity, so it will be attached. This red will be eluted out. So this step we can call it as a washing because our component of interest will be this blue color. Okay, then so some of the thing, I mean, next step will be elution. So here, this here the affinity towards exchanger resin and blue will be higher. So we have to break this affinity, break the interaction. So the higher affinity towards this cellulose matrix compared to this blue color compound that will be used here for elution. So now the elution will be there. So you can able to get the blue color fragment separately. Again, next will be fifth step will be regeneration. Again, so initial stage it will be there, no? So likewise, it has to regenerate. Okay, that is the thing we'll be doing here. So now I'll show one video that will clearly detects that clearly tells the steps easily in an animated form. Okay, I'll show this. So here the matrix will be there that will be of specific charge. Then our component of interest will be, see here you see matrix will be positively charged. So different negative charge components will be in the sample. So different color you can able to see green color, purple color and red color. So red color will be and each one having different affinity. So red color will be less affinity compared to all other two. Okay, so that will be eluted first. Then next will be green color. That will be compared to purple, the green color will be less affinity. So it will elute second. And highly affinity towards the exchanger resin will be this purple color. That will come last. Okay, you can able to see. And able to. So the profile also it will be coming there. It's a chromatograph. Okay. So next steps will be there. So majorly four steps will be there. One is equilibration, sample application across, elution, regeneration. These things already I have shown in the diagrammatic representation. So here in equilibration, so ion exchanger resins will be there that will be equilibrated with a buffer which will contain Na plus and Cl minus ions. This is the first step. Then second step will be sample application and washing. So in sample, there will be different components. So our component of interest compared to our component of interest, other things will be the waste only for this process. So these are all the things has to be removed. That will be in the washing. Okay. Then third will be elution. So here our component of interest will be tightly bound with the resin. So that has to be removed. That step will be elution. Then last step will be regeneration. So here this elution, no? So highly affinity towards resin will be used. So that will be eluted. That step will be called a regeneration process. So that I'll be showing in this diagram. So already I have shown this CM cellulose cation exchanger, no? So here it will be attract with the positively charged group. So these are all the negatively charged group. This will be through in washing step, this will be come out. So now you can able to see the in this step, CM cellulose cation exchanger will be bound with positively charged protein. So if next step will be you need to remove the specific positively charged protein. Okay, so here this compared to this positively charged, some other buffers which will be having Na plus Cl minus ions. So these buffers can be used as a elution buffer. So now while running this buffer, you can break this interaction and you can able to get our component of interest separately. So in this way, this experiment can be performed and you can purify our component of interest. So this will be clearly shown in this diagram. You can able to see first column will be packed with the exchanger resin and this low salt elution buffer will be used for equilibration. Next is sample application. Then so in that sample, what are all the different component of interest will be there? No, highly affinity, less affinity towards our resin. So that will be attached to that. Then buffer will be used for washing purpose and which will be having higher affinity that will tight bound and others will be lessly bound that will yellow come. So compared to this blue, purple and red will be less affinity, it will come first. Then next continuous elution will be there. Then the in blue and purple will be less affinity that will come next. Okay. Then third will be higher affinity that will come out last. So you can able to see in this chromatogram protein concentration versus this now. So here, out of these three things, the blue will be highly affinity towards this resin. So in this way, the components in the mixture that can be purified with the help of ion exchange chromatography. And some of the common ion exchange resins will be used is the stationary phase. I have already told CM cellulose and DEAE cellulose. Other than that, P cellulose, Bioret 70, DEAE Cephadax. Cephadax also that can be used as a base material here. And CM Biogel. So these are all the some of the readily marketly available resins will be there. Okay. So this can be bound with which type of molecule that will be shown here. So the nature of this resin also either basic or acidic that will be shown here. Okay. So this will be the specifically in this table you can able to see. So which type of compound will be attached with the base compound? So it is water. So TEAE cellulose I have already told no. So diethyl amino ethyl group. This will be with anion. Okay. So this will be the positively charged. So it can attract negatively charged proteins. So like with CM carboxymethyl, it will be negatively charged. So it will attract positively charged protein. So it will be specifically these functional groups will be active in specific pHs. That is also I have mentioned here. So now last part will be advantages and the disadvantages of this ion exchange chromatography. So advantages will be specific type of purification. So it will be specifically charged interaction will be there. So this will be a highly efficient or finest purification step we can also take. Then this can be used for both analytical and preparative purpose. So that will be one advantages. And what are all the inorganic ions? No, that also can be separated with the help of ion exchange chromatography. And the limitation will be the buffer requirement will be till be of varying. Not only one type of mobile phase we cannot use. Okay, so different steps we can use different types of mobile phase. Then preparation will be tedious. And here, so here one type of charged molecule only specifically we can separate. On other types, if your component of interest is having a 2 to 3 negatively charged proteins. So here there's a the purification will little bit tedious. But it is the limitation of this type of chromatography. And cost wise, compared to other type of chromatography, the ion exchange resins you have to prepare it and you have to pack. That is on limitation. Okay. So I think we have completed ion exchange chromatography in detail. So if you have any doubt, you can ask me. Okay, ma. Thank you.